My name is Vincent Moser. I'm a physician trained in internal medicine and board certified in internal medicine. I joined McGill last year, and the mandate I have is to build up a genomics medicine program at McGill within the framework of the Canada Excellence Research Chair in genomic medicine. My background, apart from being an internist, a clinician, is I've been training in genetics, in pharmacology and cardiovascular diseases. I've spent 10 years in industry. I've been back to Switzerland, where I come from, to lead large efforts in laboratory medicine. And here I am now in Montreal. What I'm planning to do today is first thank the organizers for giving me the chance to present this biobank, which we believe is an important asset for Quebec and also for the COVID-19 overall research community. It is also an asset which could be very valuable and will benefit from AI investigations. The second thing I will do is present to you this construction. If you don't mind, I will spend 15 minutes or so presenting that, and then we have times for questions, and I will read the chat at that time. So I'm not encouraging you to use the chat right now because I will not read it during my presentation, but then after completion of my presentation, I'm happy to have a look at the chat and identify questions. Okay, I'm starting. This slide shows on the x-axis the time, days, and on the y-axis the number of people who were hospitalized for COVID infection in the regular wards in orange and in the ICUs in burgundy. The big arrow here illustrates these dates when I met the head scientist of the province, Rémi Quirion, and Carole Jabet, the head of the Fonds de Recherche en Santé du Québec, and Daniel Coder, the head of Genome Québec. We had this discussion. It was not around COVID, but of course, COVID came up. We were 19th of March, and they said, okay, we need to put in place an infrastructure. We don't have yet any cases of patients hospitalized in the province, but we anticipate the tsunami will come, and we need to get ready. And they gave essentially me the mandate to build up a structure whereby researchers in COVID would have the necessary samples and data to do their research. The mission we were given was to ensure that scientists have access to the biological materials and data which would be needed for their research efforts, more specifically on the determinants of COVID-19 susceptibility. That means why do some people develop COVID and some others not, severity, and outcome. And in that way, we wanted to respond effectively to the public health challenges which was posed by the pandemic, and of course use sound scientific basis, and most importantly as well, an appropriate ethical and legal framework. This slide here shows you the milestones. Again, 19th of March, we received the mandate. 10 days later, we have a task force in place. So with the help of Carole Jabet, Rémi Quirion, Daniel Coder, we put up a group of 12 people, each with specific expertise in genomics, ICU, clinics, virology, vaccinology, immunology, law, ethics, communication, finance. And we work extremely hard for 30 days to first identify what are the questions we want to enable, what is the design of the biobank we need to put in place to help our researchers address this question, submit the filing for ethics approval. And we did that in 10 days so that one day later, we could start the recruitment of the first patient here at the Jewish General Hospital. We could essentially surf on this first wave so that within 2 months, we had already 1,000 patients. And Public Health Agency of Canada, which was sensitive to the fact that Quebec and Montreal in particular was very badly hit by the virus, gave us additional funding to expand our program. We did the first genetic analysis on the first 400 samples already in July, and we had the Genome-Wide Association study data available for researchers by mid of July. I just want here to list the people who have been working extremely hard to make it happen. I'm chairing this task force. We have here the name of the people, their affiliation. We have now met 62 times over the past months for an hour and a half, always by virtual meetings. I've never met half of these people just by Zoom. And we worked extremely hard together. We have put as well a governing structure. This is not exciting, but this is important within the framework of our discussion today. There is a steering committee here, which I'm chairing as the director. We have a governance committee, which is advised by an international scientific advisory board, which is chaired by Antoine Flau from Geneva. Importantly enough, as well, in our discussion today, for accessing data and samples from the Biobank, we have a data and sample, sorry for the S, access committee here, which is linked to a separate entity called Réseau Québécois COVID and Pandemic. The vision is shown here. At the bottom, you can see 11 hospitals, so it's a province-wide, federated, decentralized infrastructure, each hospital feeding data and samples into the Biobank. We managed to have 11 institutions from 5 universities signing the same interinstitutional agreement. That was already by itself a tour de force. We're now collecting samples and data from these hospitals, and the idea here is to perform an in-depth multi-omic analysis on the samples which are being collected here. Keep that in mind as well when we're thinking about AI usage for exploring these data sets. So we're working with an initiative called Cancogen to access the genome virus data. We're working with Cancogen as well to have access to whole genome sequencing data from the host. We're talking here about 3 billion base pairs for each individual. We're working with the SHUM here to get deep serology analysis and also for transcriptomics. That means analysis of the gene expression in the white blood cells collected from the blood to see expression of genes, also importantly, genes involved in immunology. And we are doing also proteomic analysis. In this particular case, we have a company called SomaLogic, which is performing a test called SomaScan, whereby they are for us measuring 5,000 different samples in the blood that we have shipped to them. What we're expecting from this data set is to better understand mechanism of propagation, individual susceptibility to diseases, mechanisms of disease protection, but also build up a cohort that we will be following prospectively. And at the end of the day, really what will count is how much this data set made available for the research community will help our stakeholders and public agencies for designing the exit strategy, optimal surveillance, hopefully participate to discovery of new drugs and vaccines, but also a patient cohort. The design is extremely simple. It's shown here. Initially, we focus on severe cases. That means people who have been hospitalized for suspicion of COVID disease. We are collecting data at admission, day 0, then prospectively for those who stay in the hospital at day 2, 7, 14, 30, and also post-discharge up to 24 months. We are also now starting to collect patients who have not been hospitalized. That means moderately or mildly affected individuals. A unique feature of this biobank is that we are collecting controls as well, which are critical for any type of biomedical trials. These people are controls. That means they have a suspicion of being COVID infected, yet PCR is telling us that they do not have SARS-CoV-2 infection. This is the network of the hospitals which we are working with in the 5 universities. Of course, again, I need to acknowledge here the very goodwill of all these people. We have 11 lawyers talking to each other to come up with an agreement that makes everybody more or less happy. The recruitment process is standard. We are collecting the consent of the patients, which is particularly complicated when you're dealing with highly infectious people. Same thing for the handling and collection of biological samples. This is infectious material. We need to be very careful about protecting the health of the working collaborators. And the data collection, extensive clinical data, also access to medical records, laboratory data. And so that's a big body of data which is being made up and will be made available to researchers. We're talking here about probably 300 people with a variety of different professional expertises and who have essentially rolled up their sleeves to make it happen. Clinical research, nursing care, computer, laboratories, administration, legal, ethics, and technical services working together to make it happen. As per the end of the wave 1, we had 1,200 participants. Now we have 1,500 participants who have been recruited in this study. And we are in the process of doing the initial analysis on them and make them available, as I said before. We have whole genome data for 500 people, and this data will be made available very soon. Actually, it's on the website. I just want here, because we're talking also use of AI and big data for drug discovery and development, to show you this particular slide. It is a project run by Dr. Brent Richards at the Jewish General Hospital. And the idea here is to use a particular bioinformatic complex approach called Mendelian randomization to identify new drug targets for COVID-19. And what Brent and his team came up with is by combining very big data sets from the UK Biobank and other data sets and genome-wide association study for COVID, come up here with 4 different molecules, circulating proteins, which are closely and very significantly associated with COVID susceptibility, severity, and hospitalization. And they may represent potential drug for repositioning for COVID indication. So what are the next steps for us? Of course, we want to continue the recruitment of severe patients and also accelerate our recruitments of outpatients. So far, we have 250 people, outpatients. We want to follow these individuals longitudinally, provide data and samples for researchers, link this data with other national and international initiatives. Of course, disseminate the knowledge developed to public health authorities and the Canadian public, and develop interfaces with the scientific and patient community, in particular through the website, which is shown here. So on this website, there are also conditions and ways by which researchers can access the data. So I'm done with my presentation, and I don't know if there are any questions. We have a question here by Jackie. What kinds of data points are we collecting for the patients? So we are collecting serially data and samples. For the patients who are hospitalized, this is the study design. Now, we do not have every time points for every patient because it's very complicated. We have, for a certain fraction of patients, 0 to 7, we are starting as well to have these data sets after discharge. We are also facing difficulties in the sense that due to the pandemic and the sanitary measures, it's very hard to go back to people, who convince them to come back to the hospital for blood draws. So we're designing now new ways to get these data in samples. That's one thing. The second thing is that there are lots of patients who are essentially saying, we've been sick, it was a very severe flu, sometimes we've been in the ICU, but it's over. We don't want to talk about it anymore. So we have to put in ways as well, modes for essentially keeping these people and reduce attrition. Our website will give precisely the number of people who have been collected at different time points. Jackie, does it answer your question? Stephanie, you're asking, will we be able to study the long-term effects of COVID that we see in some patients? The answer is yes, we are planning to. And actually, we have funding as well to follow up these patients up to 24 months. I think the question is absolutely essential. I can just refer to some preliminary data, which I have seen from the UK, a program called See More, whereby they have prospectively followed patients in controls, actually, and they show, using imaging and other quality of life measurements, that indeed COVID-19 infected patients, at least when they were reassessed after 2 or 3 months, have a higher probability of having anxiety, depression, but also multi-organ inflammation. So it's a very good point, and we will do our best to get as many patients as possible. We were lucky because we could capture this first wave. Not that many hospitals could capture first wave patients because they were so busy in doing clinical care. Urs, you're asking the question, how does the information exchange work across the provinces and internationally? This is also a really important point. So here we're committed to open science, firstly. And so the data in the samples, yes, that we are collecting are being made available to the entire research community. So we have this data and access committee, which has as a remit to make sure that these data and samples are made available. The consent, and that's really critical, allows for sharing of the data for researchers, including industry, internationally as well, as long as the researchers adhere to the standards that we require. Now, we have already started to share the GWAS data, the Genome-Wide Association study data, and we are part of this genomic host initiative. And so we are already contributing data to these efforts. Jackie is asking for a comment on my response. Clinical data. So the clinical data are part of the data which are being made available to the research community. And we are in the process of putting in place the IT infrastructure with Michael Chassé at the CHUM and with Genome Center, Guillaume Bourque, so that this data is properly packaged for sharing while protecting the identity and avoiding re-identification. Isabel, is asking about the correlation between ABO blood group and susceptibility to COVID-19. I can elaborate. I'm not sure I have the response, but what seems to happen is that this signal with the ABO blood group may be, and I'm saying really maybe, be careful, may be an artifact. It may be due to the fact that this New England paper was based on data generated in Italy and Spain mostly, and the controls were people from blood banks. And we know that people who are donating blood at the blood banks are not representative of the general population, because they are also asked to donate blood based on specific blood groups. So it is possible that we may have an artifact. That's also an incentive for us to recruit and collect data and samples from our proper control SARS-CoV-2 negative populations. From what I have seen in the recent metadata from large consortia, the ABO group signal seem to vanish. Stéphane, tu demandes what more immediate use will be made of the biobank by AI or digital projects and approaches? There are a series of things that we could do with AI directly with our connections, the biobank and, for instance, Mila, and we have already been discussing with you, Stéphane, and with other people at the Mila. Using this data set and see how AI can be used essentially to exploit it and maybe identify gems as well and jewels hidden in this mountain and address some specific questions. We're also working with closely with Michael Chassé. Michael has a CIHR grant for using AI in collaborations with Mila and others on COVID data set. Finally, as I said, the data will be made available for investigations around the world who can use essentially their tools, including AIs, to explore this data set. I don't know if there are any other questions. I think there is one from Nathaniel. Yes, Nathaniel, you refer here to a paper published by Jean-Laurent Casanova and his team in the Science issue around 3 weeks ago now. And what Jean-Laurent Casanova and his group did was investigate using sequencing 13 genes involved in the interferon pathway. And you're right, they identified some mutations which seem to predispose to disease COVID severity. Have we found the same? I cannot answer this question yet because we are just now QCing the latest whole genome sequencing data, but we will certainly look at that. What we have done though in my group is look if some of these mutations, they are rare functional mutations. If these mutations are present in excess in the Quebec French-Canadian population, because this French-Canadian population has a founder effect, and so some variants are overrepresented. What we have found, and that's the work that Claude Béret has done in my group, is that indeed some of the variants identified by Jean-Laurent Casanova and his team are overrepresented in our Quebec population, and we need to explore that further. If there are people interested in this work on rare functional variants and how we want to exploit them for supporting drug discovery and development, I'm actually giving a second talk in an hour. I think it's at 3:50. That's what I have presented here is my sort of moonlighting job. And I have a day job, which is this Canada Excellence Research Chair in Genomic Medicine, which I'm presenting a bit later. So I guess if there are no more questions, I would like to thank you all, 23 attendees, it's great. I'm very glad I had the chance to present this work, and I certainly look forward to having this wealth of data, including multi-omics data, that will be available for researchers and for AI exploration. So I will stay in touch certainly with Mila. I will stay in touch as well with industry, because this biobank also serves as a reservoir of patients for trials. And we can now also use AI and other markers, including genomics, to potentially select patients for trials and deep phenotyping. So thank you very much to all, and it was a pleasure to share that with you. Bye-bye.